From 2 nanometer, its thickness or diameter has become 10 nanometer. Now it again undergo further coiling to form solenoids. Remember, so each solenoid, one turn of solenoid will carry 6 nucleosomes. So it starts turning. First of all, it coils around this ball-like structure. Then it starts turning around to form six nucleosomes together to form one turn. What is solenoid in physics? We have already done a spring-like structure with a hollow thing inside. So it is hollow inside. Space is occupied. That means what? A 2 nanometer thick structure turns into 10 nanometer. Now it becomes 30 nanometer. So diameter is increasing, length is decreasing, and it further changes into supercoils, which will be somewhere around 300 nanometer. Please remember, chromatin is going to be 10 to 80 nanometer in its diameter. Chromatin, I forgot to mention this. And this 300 nanometer thick structure is the super solenoids, which will further undergo coiling to form chromatid. Chromatid is, if you remember, is the half portion of the chromosome, which you have learned even in the 11th class. So chromatid is 700 nanometer in its thickness. And we all know chromosomes are duplicated amount of DNA, so you can call it as 1400 nanometer, because double amount of chromatin. 1400 nanometer is the diameter of the chromosome. So as the diameter increases or thickness increases from 2 nanometer to 1400 nanometer, there is a huge reduction in the length of the genetic material. I hope this whole concept is clear to you. Nucleosome and solenoid model of the folding and supercoiling of the basic chromatin components to form in the case of this. So eukaryotic packaging is quite complex when we compare it with the prokaryotic one. I hope this is clear to you. Now the next thing to understand is the packaging at the higher level of requires additional. So remember, when this packaging is going on, there will be further proteins required which are non-histone chromosomal proteins, and they help in the formation of chromosomes. So those proteins are called as non-histone chromosomal proteins. They help in the higher level of packaging and coiling of the genetic material to form something called as chromosomes. In a typical nucleus, now when you see the nucleus of any eukaryotic organism, you will find its genetic material during interphase. Please remember, interphase is the stage when genetic material is like a thin loosely arranged thread-like structure. So all those thread-like structures are chromatin, and I just told you a few minutes back, the chromatins are 10 to 80 nanometer in their thickness. Why so much variable 10 to 80? Because there can be areas where thickness is less, and then there are areas where thickness is more. So let's understand what are those two areas called as. Now the next thing to understand is that in a typical nucleus, some regions of chromatin are loosely packed. So loosely packed means 10 nanometer, and they will stain light in color because they are thin areas. So when you put stain or dyes, like for example, if you put the acetocarmine, you will find the staining is light. That means color picked up is very light. You call that area of the chromatin as euchromatin, and it is the area which is transcriptionally active. So that means euchromatin participates in mRNA formation and protein formation. The other region that would be 80 nanometer in thickness. So why it is thick? Because it is densely packed, and because it is densely packed, it will pick up the stain also in darker color. So those areas will stain darker and are called as heterochromatin. And remember, this portion of the DNA is inactive in nature, so it is not involved in the transcription. So it will not be causing the formation of mRNA and protein. Please remember, heterochromatin can also be of two types. I have forgotten to mention in the notes: constitutive and facultative. What is constitutive? There are some portions of the heterochromatin which are never going to lose. Because they are not going to lose, they will never become euchromatin. So that means what? Heterochromatin can be of two types. Some of its portion will always remain non-functional, so they are constitutive, and some portions may become active, or they may become loosely packed, or they may become transcriptionally active, and they can be called as euchromatin for some time. That's why they are called as facultative. So heterochromatin can be always constitutive type, or they can be areas where they may lose the property to function, again gain the property to function according to the physiological state of the cell, or you can say the nucleus gives the command which portion should function, which portion should not function. So accordingly, heterochromatin amount varies inside the cell. It may, but there are some portion which never function, and they are called as constitutive portion of the heterochromatin. Remember, histone proteins are rich in these two amino acids mentioned in the NCERT. That remember, histones are positively charged. They are positively charged because of the basic amino acids which are there, and histones are rich in two basic amino acids, and the name of those amino acids are lysine and arginine. In 11th class, you have learned the complete list of the amino acids. Just check there. Lysine and arginine were explained by me in the class as the two examples of the basic amino acid, lysine and arginine. I hope this whole concept is clear to you. Let me quickly show you what exactly NCERT says about this topic later. So the whole concept of NCERT has been done, and the last thing to understand here is the same non-histone chromosomal proteins are there, and then two portions of the chromatin are called as heterochromatin and the euchromatin. So in the next class, we are going to start with the topic that is the search for the genetic material, and we will be learning about some experiments performed in the history which made clear to the scientists that DNA is the genetic material.